I've opened a lot of letters and emails in my life, but only a few have touched or humbled me. Let me share one of them with you. Dear John, I was reading about you, and it occurred to me that I should send you a personal and heartfelt note of thanks. I'm not smart enough to have any idea of what you were doing when you invented Somavert, but I am smart enough to know that your work and Somavert have enabled me to have a normal and much longer life that I would not have had without you. I don't know how to start to say thank you, but I know that I should try. So thank you. I will forever be grateful to you for what you've done for me and other patients with acromegaly. Sincerely, Jim. I have to tell you, I get chills every time I read one of these letters. In the mid-1990s, Jim was diagnosed with acromegaly, a devastating, disfiguring disease caused by a tumor in the pituitary gland at the base of the brain behind your eyes. It produces too much growth hormone. Andre the Giant is probably the most famous patient you might know with acromegaly. Growth hormone, as its name implies, is important in controlling growth by increasing bone, increasing muscle, and decreasing fat in your body. These are some of the good actions of growth hormone. But for people with acromegaly like Jim, too much growth hormone causes uncontrolled growth, resulting in extreme pain in the bones, the joints, and even in the teeth. Their skin becomes thick, and ultimately their hearts, liver, and kidneys are affected. Left untreated, acromegaly leads to premature death. There are about 100,000 people worldwide with acromegaly. Additionally, too much growth hormone is associated with elevated risk of diabetes and cancer, and patients like Jim often come down with these disorders. Somavert, the drug that helped return Jim and so many patients like him to a normal life, was born out of my fascination with growth hormone and its positive and negative effects on our bodies. The story behind my lab's discovery, from compound to market success, is one of big questions and even larger goals, along with the foresight to protect our scientific results and sprinkled in a little bit of serendipity. I'll start the story in the late 1986 as I was getting ready to move to Ohio University, OU. In truth, I thought I was going to the Ohio State University. I had not heard of Ohio University. OU was setting up a biotechnology institute and needed someone who was a card-carrying molecular biologist to lead a team of investigators. At the time, it was hard to leave my work at Merck and to go to academia. I was happy at Merck. I was productive, I liked the people, and they liked me. But I could not resist the mission of OU. The objective of the Edison Biotechnology Institute at Ohio University was to do research, obtain grants, train graduate and undergraduate students, hire postdoctoral fellows, develop internal, external, and international collaborations, and if the results looked like they could help humanity, patent the findings. The hope was that these discoveries could ultimately get out into the world where they could help people. Now let me tell you folks, these are pretty lofty objectives. I was given the mandate to hire 4 individuals who would help me develop my scientific team. My early experiences had taught me just how important it was to work with good people who ask good questions. Back in the early 1980s, as a postdoctoral fellow at the Roche Institute of Molecular Biology, I had the honor of working with some of my scientific heroes. I can clearly picture our casual daily lunches, just 5 to 10 postdocs with a Nobel Prize winner and 3 National Academy of Science members talking about baseball, football, and if you can imagine this, the joys of eating vanilla pudding. And of course, we talked science, a lot of science. What struck me most was the way these heroes asked questions. So many good questions. To them, no question was off limits. So when I established my research group at Ohio University, I formed a large lab family. I taught them to be courteous, kind, work as a team, and don't ever be afraid to ask questions or take on the big projects. We set out to do some molecular biology experiments on growth hormone. At the time, I also wanted to help children who did not produce enough growth hormone. These children were short in stature. The only treatment for these growth hormone deficient kids was a drug called recombinant human growth hormone. However, the drug needs to be injected daily, and in some patients, this could take daily injections for over 15 years. No one likes needles that much. Even more critically, the additional growth hormone posed a risk of diabetes and cancer. We wondered, could we change or mutate the growth hormone gene and remove its bad actions, that is diabetes and cancer, while keeping the good actions: less fat, more muscle, more bone. Also, we hope to generate a new growth hormone that would last longer when injected into the body, so that these short growth hormone deficient kids will only need to have the shot perhaps once a week instead of daily. This was truly a big picture project. Where to make the changes in the growth hormone molecule was a challenge, since it has 191 units or amino acids. We focused on one small region of the growth hormone molecule, a region called the alpha helix. We had reason to believe that one amino acid in that helix was crucial for growth hormone's actions. I assigned a graduate student to change this amino acid, hoping that the resulting molecule would be a better growth hormone. All of the good of growth hormone and none of the bad. And maybe as icing on the cake, it would have a longer half-life in the body for those growth hormone deficient kids. We already knew that when extra copies of growth hormone are inserted into mice, one gets large mice with little fat, big bones, and muscle, all good actions. But these animals also develop diabetes and cancer, bad actions. So for our growth hormone molecule with one amino acid change, I expected a really giant mouse with big muscles and bones, little fat, and no diabetes or cancer. I convinced my graduate student that this had to be what we would get. I could not wait for the results. But they were slow, very slow to come in. It turns out that the student was afraid to tell me the results. He was getting small mice instead of giant mice. When he finally came to me with results, I saw a small mouse. I said, "What? I don't believe it. Something happened. We got to repeat the experiment." So I went overboard in my enthusiasm for a giant mouse and didn't really want to hear about a mini mouse. But sure enough, when he had repeated the experiment, we got small mice. It was a bit of serendipity. Instead of a growth promoter, we had discovered a growth inhibitor. I knew immediately what we had to do. You see, on my first day of my first real job at Merck, the president of research told me, "Kopchick, I want you to work 100% of your time on Merck projects, 100% of the time. With the other 50% of your time, get it, moonlight on anything you like. But if you happen to discover something that is an inhibitor, immediately go to the Merck lawyers and patent it." After all, many of the big-time drugs are inhibitors, including statins, which were discovered at Merck. Perhaps some of you take this drug for lowering your cholesterol. So we had an inhibitor and started the patenting procedure. During the process, we figured out how the new compound worked. It was a growth hormone receptor antagonist. That's a biochemical name. We received a patent on the molecule, and after many setbacks trying to get large pharma to license it, I couldn't get anyone, we set up a small company with an OU alumnus. Another case of serendipity. After approximately 10 years, we ultimately had a compound approved for use, and it's now marketed worldwide for patients with acromegaly. Remember, these patients have too much growth hormone. We're very proud of how a little mouse ultimately resulted in a drug. We did translational research, that is, from laboratory bench to patient bedside, before it was called translational research. But for me, it's all about the people like Jim, who we have helped. And we're not done asking big questions. I embrace big questions. Right now, we have a new lofty goal. We want to cure several types of cancer. Pretty lofty. A variety of human tumors grow in the presence of growth hormone. Pancreatic cancer, melanoma, liver cancer are 3 examples. So with a Columbus-based company, we have come up with a better growth hormone antagonist and wondered, would it inhibit tumor growth? Remarkably, it did. The results so far are very exciting and promising. Also, unexpectedly, our models confirm that when the new growth hormone receptor antagonist is mixed with current cancer chemotherapies, this cocktail or mixture can entirely eradicate the tumor, a cancer-free state. Let me repeat that, a cancer-free state. A finding of this type could positively help many, many patients. Imagine a future where cancer patients can take less chemotherapy in combination with a growth hormone inhibitor and become cancer-free. Lofty goal. So we're once again in the process of taking our work from the scientific bench to the patient bedside. I know that if we stay focused on this big, audacious goal, protect the results along the way, and leave a little room for a bit of serendipity, we can make this dream a reality. Thank you.